Today we're going to be continuing on our discussion of populations. And so I thought I'd open by showing you a photograph of a brown booby, which is a kind of a seabird, flying along a sheer cliff face back to its breeding colony. And in fact, here is the breeding colony, the base of that cliff in coastal forest in the tropics. What you're looking at here are not brown booby nests. This is a different species, however. This is the blue-footed booby, white bird with powder blue feet. And there actually is a third species that nested in this colony as well, the red-footed booby, which indeed did have red feet. So a nesting colony is, you might say, ground zero in terms of where population growth begins. Depending upon the success of breeding, that will determine whether or not populations are growing or shrinking to a significant degree. There are, of course, other factors, as we've discussed in previous episodes, that influence population size, growth and contraction as well, but certainly breeding success is a big one. So let's begin where we left off last time, talking about how it is that we might go about estimating population size. As we began discussing last time, one of the traditional ways of estimating populations of wild animals is to mark them. And so that's what you see in this photograph here. We're looking at a nightingale reed warbler, endangered species, by the way, and we'll come back to endangered species toward the end of today's discussion. But this individual here, you see, has colored bands around his right leg. On his left leg, which you can't quite see in this photograph, he also has an aluminum band with a number stamped into it. The color bands allow us to tell which particular individual this one is. Those color bands are actually visible in the field through binoculars, and so we can keep track of individual birds with those color bands. They also are often coded so that we can tell males from females. So usually if I put a blue band on a bird, that means it's a male. And usually if I put a yellow one on a bird, it's a female. Here is the actual process of applying the band. The bands themselves actually have a split in them so that they can be opened up and then placed around a bird's leg. And then using these special pliers with this little opening in them, the band can be squeezed tight around the leg without having any possibility of damaging the leg, because the band can only be squeezed so tight with these. And so once a bird is banded, now we have an individual within a wild population that's marked. And if we capture a bunch of these and mark them, now we have a proportion of the population that is banded. And so we can use some statistical kinds of techniques to estimate what the total population size is from the percentage that we have banded. The simplest method for estimating populations from marked individuals is called the Lincoln-Peterson method. And it's simply a ratio and proportion problem. If we know how many individuals are marked, and then we go out and recapture individuals and look at the proportion of the recaptured individuals that are also marked, then we can come up with an estimate of total population size. This is a very simplistic method, however, that doesn't take into account many of the kinds of things that go on in nature, like individuals dying, individuals moving into an area or leaving it, the fact that over a period of time we might expect to see reproduction and addition of individuals to a population. those kinds of things. So in statistical language, the Lincoln-Peterson method is what would apply to a population that is closed, no individuals moving in or out. This is the method that is usually discussed in AP environmental science courses, although in reality, few real scientists use it. It's really only applicable to very short-term studies where you're going out, capturing individuals, and then over a period of a couple days doing the recapture. Most capture, recapture kinds of experimentation uses this next method called the Jolly-Sieber method, which is rather more complex to apply, but fortunately there's computer software that will take care of all the computations, and this is available freely from U.S. Fish and Wildlife Service on their website. The Jolly-Sieber method is one that can be used for populations that are being studied over a longer period of time, when individuals can be expected to be moving in and out of an area, when you can expect that some individuals are going to die for one reason or another. And if we're studying a population over the course of a year, then we can also expect that reproduction is going to be occurring, so new individuals will be added to the population. Even using this much more sophisticated protocol, however, there are certain assumptions that underlie these sorts of things. One is that your sampling of a population is truly random. And that's not necessarily always the case, because individuals, once they've been captured, can become trap shy. They become wise to the idea of how you trap them, and so they go about avoiding traps. Other individuals sometimes will start becoming attracted to the trap itself, and they'll get trapped over and over again, so they'll be disproportionately trapped. A second assumption is that the fates of individuals and age classes are independent of each other, and that is also not necessarily true. Juveniles, for example, often suffer differentially high mortality compared to adults, which is not surprising when you think about it. They are more naive about predators and less adept at finding food and things like that. A third assumption is that there is no loss of markers on marked individuals. This assumption does tend to be closer to the truth than the first two, actually, in most kinds of mark recapture studies. Now, there are other kinds of markers that can be applied to animals other than bands around legs. That's pretty much for birds. In the case of mammals, often ear tags are used. In the case of reptiles, sometimes their scales are painted. There's all kinds of different kinds of techniques for making markers. In the case of turtles, sometimes little notches are put in their shells. Let's focus on this species, the bridled white-eye, which is a tropical island bird found on just a few islands in the tropical Pacific. This species, I made a number of different approaches to estimating its population. It was an interesting species to study because its densities were among the greatest ever recorded by any bird species anywhere on the planet. So I looked at population densities using several different techniques, including Jolly-Sieber and some other methods too that we'll talk more about later. But anyway, in order to do Jolly-Sieber, I had to capture and recapture individuals. And that's what you see going on in this picture here. Bridled white-eye is a flocking bird, and so when I set up a series of nets that I could elevate into the canopy of the forest, and these species do live up in the treetops, I could catch a whole flock at once. So sometimes I would catch 7, 8, 10 birds at the same time. One of the side benefits of capturing birds was also that I was able to learn to distinguish between adults and juveniles. If you look at these two birds here, the one on the left is a juvenile, rather gray, just subtly grayer than the other bird, which is an adult with more yellowish tones in it. And that allowed me to learn something about population age structure. So in any event, once I captured and recaptured birds over the course of the better part of the year, I was able to apply the Jolly-Sieber method to estimate populations. But I also did something else concurrently while I was capturing birds. Every bird that I captured and marked, I also color marked with those color bands that I showed you in a previous photograph. So once I let birds go, I was able to observe them through binoculars and see where they went. And by exploring an area of several hundred meters around the banding site in this image here is represented by a long black rectangle. And if you look at those little black squares, those are resightings of birds. And so I was able to plot those on a map and then examine what the plot showed me. By the way, in this image, the larger black boxes are buildings, as are the gray rectangles. So if we just look at those little black squares, those are the resightings. And when I plotted them and examined them, what I discovered was that the further away I got from the banding site, the fewer marked birds I encountered and the more unmarked birds I encountered. And I was able to demonstrate that there was a rather precise mathematical relationship with how the number of banded birds declined with distance. In fact, it fit a quadratic model. If you ever wondered why you learned all that stuff in algebra, this is why. Because you can use those equations to model phenomena that are observed in nature. And so once I had this quadratic relationship demonstrated, I could use that relationship as a means of estimating population size. So I was able to compare that estimate with my Jolly-Sieber estimate as kind of a check on whether or not these two estimates were giving about the same kind of result. And as it turned out, they both were in reasonable agreement on population densities. Now, I use still one other technique to estimate populations of this species. And that's what I'm doing in this photograph here, is I am out censusing individuals rather than capturing them. So recording where I found individuals at survey points. So this is an observation-based method of estimating populations as opposed to a mark recapture kind of a method. Observation-based methods of estimating populations may be either relative or absolute. Relative kinds of methods involve going out and counting individuals but not being able to use the data that's gathered to actually estimate actual densities or population sizes. Absolute methods do allow for that, and that's actually what I was doing with my method. I wanted to have an absolute population estimate that I could compare to my mark recapture methods. Examples of relative kinds of population estimates are things like the U.S. Fish and Wildlife Service Breeding Bird Survey and the National Audubon Society's Christmas bird count. This map that you're looking at here is actually generated from breeding bird survey data. However, the data are relative data. They simply involve driving along a route and getting out periodically and counting birds. And so from year to year, you do get some relative idea of whether or not populations are going up or down, or how one place compares to another in terms of numbers of sightings. But you can't estimate birds per hectare or birds per square kilometer or whatever using these kinds of techniques. A much more sophisticated kind of observation-based population estimation is what these people here are doing, which falls under the heading of what's referred to as distance sampling. The particular technique that they're using is called the variable circular plot. And a variable circular plot involves setting up sample points along a transect, usually at about 200-meter intervals, far enough apart so that individuals encountered at one sample point are not again counted at the next. That's called sampling independence, by the way. And then individuals that are encountered at these sample points have the distance between them and the observer estimated. And then based on that kind of data, actual density estimates and total population size estimates can be made. It's a rather kind of a difficult procedure to employ, so usually only professionals, people with a great deal of experience in this sort of thing, make use of this procedure. So once again, absolute population estimates are those that are involved in estimating actual population density in a technique that does not involve capturing and marking individuals. Now, the technique that I've been discussing is the one called the variable circular plot. But there are other techniques that exist. In addition to the variable circular plot, there are fixed width survey transects that are used in certain kinds of situations for certain kinds of animals. Now, one of the things that the variable circular plot generates is something called a detectability function, because animal species each have their own detectability, that is, how easy they are to find. With certain kinds of birds, for example, they're loudly vocal and they are frequently vocal. Other species are rather quiet and just slink around, so they're harder to detect. And so in order to estimate populations, one has to have some information about their detectability. And through the kind of data that are gathered in the variable circular plot, it is possible to generate graphs that demonstrate how the probability of detection changes with distance. And there's actually computer software which is available for doing this, because it's a rather complex calculation to do otherwise. Let's look at an example of a detectability function, and that will help to clarify what I'm getting at here. This particular one is for the rufous-sided towhee. So what we see here is that when an individual bird is right near the survey point, its probability of detection is approximately equal to 100%. But as birds are encountered further from the sample point, the probability of detecting them goes down, so that by the time you are about 40-50 meters away, the probability of detecting an individual drops quite a bit. Once the shape of that curve is known, then that can be used to generate an absolute density estimate. If we perform these variable circular plot surveys at a number of different geographic locations, then we can do something additional with the data we generate. What we're looking at here is a photograph of a veery, which is one of the most abundant forest birds in northeastern North America. And this is one of the species that I've surveyed all across southern New England to find out the distribution of its densities. And that's resulted in the ability to prepare maps like this. This is a map of the density distribution of the veery in forested landscapes across eastern Connecticut. The right side of the map is the Rhode Island border. The upper part of the map is the Massachusetts border. The bottom of the map is the coast of Connecticut. And the left-hand side of the map's border is the Connecticut River Valley. So within that region there, the dark areas are areas where the veery reaches high density. The yellow areas are moderate density, and the white areas are lower density kinds of areas. So once we have information like this for an entire community of animals, that's quite a valuable conservation tool if we know what is where and in what numbers. The first thing we always need in terms of conservation is knowing where your resources are. So let's change gears and talk more about the conservation aspects of all of this population estimation information. We're looking here at a mangrove flycatcher, which is a species native to the Palau Islands in the western Pacific. It is a member of a superspecies, that is, a group of very closely related species that occur on several tropical Pacific islands. Now, there was once a species on the island of Guam which was virtually identical to this one. And in fact, it may have not even been a separate species, but a different subspecies. But that population is now extinct. In fact, the last individual died while I was there. Some years back now. This is over 20 years ago now. The last one disappeared. In a precipitous decline, within a matter of months, in fact, it went from being a population that we thought we could save to being extinct. If we think in terms of a calculus function, when one reaches a vertical asymptote, that means that things happen with infinite speed, and that is rather like what we saw with the Guam flycatcher. It went from having a viable population to there being none left within the matter of weeks. And so that species, or that subspecies as the case may be, is now beyond help. But there are still others that have populations that are still savable, although they are in critical trouble. And those species we refer to as endangered species. And of course, one of the things we want to know about them is what is the status of their populations and what are the trends in their populations. So let's talk specifically about endangerment. Endangerment occurs when deaths exceed births, so that populations decline. And as populations shrink, they might start losing their genetic diversity. They start losing their distribution. Their distribution may contract. We might find situations where inbreeding occurs because there's so few individuals left, and this leads to inbreeding weakness in the genetics of the population. Uneven sex ratios are not uncommon among very rare species, where males may outnumber females, and so the actual breeding size of the population is even less than the total population size. All these kinds of things can impede the recovery of a population from its endangered status. Endangerment occurs when populations decline to a level at which they become vulnerable to extinction. Most endangered species are those that continue to experience these kinds of population declines. Now, there's a number of phenomena that can lead to endangerment. On Pacific Islands, the introduction of exotic competitors or predators has been a significant factor. And in fact, on islands around the world, those kinds of situations have decimated island populations of many species. Exotic diseases also can play havoc with island species that have evolved without any experience with such diseases, so that when the diseases get there, the populations have no immunity, and so they succumb to it. Things that mainland populations may have long since dealt with and have resistance to. Other factors include habitat alteration, habitat destruction, environmental pollution, hunting, and of course all these factors don't necessarily just affect island populations, they can also affect mainland populations. Islands and their populations do tend to be particularly vulnerable to such things, but certainly mainland species have also become endangered for similar kinds of reasons. Now, the bird we're looking at here on this slide is the Guam kingfisher, which is now actually extinct in the wild. It only exists as a captive bred population, although it has a very close relative again on the Palau Islands that lives with the mangrove flycatcher. And this is another kind of a situation where the one on Palau may be the same species as this, even though it's not quite the same color. Color can change quickly in populations, and so color is not alone necessarily an indication of how genetically distinct populations are. So the Guam kingfisher and the Palau kingfisher may actually still be the same species, just well-marked populations. But in any event, the Guam kingfisher is extinct in the wild, exists only in captivity now, and so it's hard to know what to do to reestablish populations back in the wild now that there is no wild population to look at. What people have been doing is looking at the Palau Island birds to see if they can learn enough from that to guide their reestablishment protocols in years to come. So on that note, we will end for today, and we'll pick up with more about endangerment and conservation next time.